Hello and welcome to the first lecture in my fourth lecture series. As you can see, this one is entitled Pharmacology, Pharmacokinetics, a title which is a mouthful, but really gives us a bit of a preview, as a good title should, I suppose, to the topic I'm going to address today. That is pharmacology broadly and briefly, and then an aspect of pharmacology, pharmacokinetics. Now, in these lecture series that I've been doing recently, we've been focusing on broad and basic issues which I think are important to understand as we consider psychological aspects of drug use and abuse. And so some of the material that I've been trying to teach in the past lectures and that I'll teach in this lecture, I think provide a good foundation for you to think about drugs, and a foundation that you can build upon when in later lectures we tackle specific drugs like alcohol or marijuana, caffeine, nicotine, etc. So by way of an overview for today's lecture, I'm just going to quickly do a review of pharmacology because, as you may remember from an earlier lecture, we've actually talked a little bit about this topic before. And then, as I suggested just a few seconds ago, I'm going to focus on pharmacokinetics. I'm going to talk really about what pharmacokinetics is and identify a few key steps in the pharmacokinetic processes of several drugs. Then, towards the end of the lecture, I'll talk a little bit about drug testing, because drug testing, it's an important issue, it's topical, it's in the news from time to time, and it's something that draws upon some basics of pharmacokinetics, so I think it's worth considering. So first off, the quick review of pharmacology. Now you may remember back in, I think it was the first lecture series, maybe the second or third lecture, no, I can't quite remember. Regardless, in a previous lecture that I did, I asked the question, what makes a drug effective or deadly? What makes a drug toxic? And oftentimes the answer to that is, well, the amount of it that is used. And I talked a little bit about dose-effect curves, such as the effective dosing curve or the lethal dosing curve. Now, these ideas, dose-effect curves and the like, are all basic parts of pharmacology. Now, pharmacology is just very broadly defined the study of drug dosing and drug effects. It's a very broad area, and if you were studying in pharmacy or in the field of pharmacology, you could spend a very long time looking at all the interesting little nuances of pharmacology. But for our purposes, I think it's valuable to break down this broad field into two subfields or subdisciplines. The first one is pharmacokinetics, and we can think of this somewhat simplistically as the study of drug dosing. You sometimes hear the study of how drugs enter into and move through the body. You sometimes hear pharmacokinetics described as the study of what the body does to a drug. That is, how a drug is processed by the body as it is in contact with various tissues of the body, as it's absorbed into the body, and as it is metabolized and excreted from the body. So pharmacokinetics, complicated word. To simplify it, we can say it's just the study of drug dosing or drug doses. Pharmacodynamics is kind of the other side of that coin, or the other side of that definition, which is the study of drug effects. And we can think about this as what the drug does to the body. So if pharmacology broadly is the study of doses and effects, pharmacokinetics is the part of that study which is focusing on the doses or the dosing, how the body is processing a drug, and pharmacodynamics is the study of the effects that the drug has on the body. And each of these subdisciplines itself can be very complicated, but I think they're both very interesting, and I'm going to try and highlight some bits and pieces of each one in this lecture and in the following. So here is just a little diagram to kind of illustrate, if it's not already obvious, that pharmacology is the broad term, and pharmacokinetics and pharmacodynamics are these subdisciplines or subfields which build into or inform pharmacology. Okay, so with that bit of a review in place, let's focus on pharmacokinetics. And as I said already, pharmacokinetics is really the study of how drugs are processed by the body, the study of how they're absorbed and distributed through the body, how they are biotransformed and eventually excreted from the body. And depending on the type of textbook you look at, whether it's a sort of pharmacology textbook or a medicine textbook, a pharmacy textbook, a good biochemistry textbook even, you'll see different steps which will be identified in the pharmacokinetic process. Here is a table from one such textbook which identifies different steps that happen when a drug enters into the body. My purpose in putting this slide or this table or list, I guess, in this slide is just merely to illustrate that it's a complex process. When you take a drug into your body, a lot of different things happen as a consequence of that. Your body processes that drug in many different ways, typically in a long sequence of steps. And you don't need to memorize all of the steps on this particular list. Just recognize, if you didn't already recognize, that a lot of stuff goes on when you take a drug into your body. And there are a lot of interesting things that we can think about when we think about drugs going into people's bodies. Here's an opportunity to do some thinking. Imagine you have two people of roughly the same age. You've got a man and you've got a woman, and they're both having the same drug. In this case, it's alcohol in what looks to be white wine, and let's assume they're both having the same dose, so about one standard glass, which may be like 6 to 8 ounces probably. Who's going to get more drunk from that dose of alcohol? So who is going to have, in essence, a stronger effect from that drug? So think about that for a second. You probably have some intuitions one way or the other, and try and ask yourself, why is it the case that one person, one of these two people, will have a stronger effect from the drug than the other? Okay, so first off, in pharmacokinetics, dosing, hopefully a conclusion you drew from that last little thought experiment is that probably these people would differ in the effects that they get from the drug. Probably, they're not always. The woman would feel a stronger effect, getting a little bit drunker than the man, maybe a little bit drunker faster than the man. That has something to do with differences in the type of dose that we're giving and the type of body that that dose is going into. Now, I said that the two people are about the same age, but probably the man weighs a little bit more than the woman. That's not always the case, but on average men tend to be heavier than women. So the man in essence is a larger body to apply that dose into. If you've taken a good biology or good physiology class, you may know that women have a larger percentage of their body mass made up of fat tissue than do men. This means that men have is in essence more non-fat tissues in their bodies, and thus a greater volume of water in their body than women do, on average. When we think about a drug dose, and in this case I said the dose of alcohol that each one is getting is the same, that same dose of alcohol is going into a larger vessel, the man, and going as compared to going to a smaller vessel, the woman. So we might expect that the woman in essence is getting more of the drug relative to the amount of available water there is in her body for that drug to go into. The man getting relatively less given the amount of water that there is in his body for that dose to go into. So at a very basic level, when we think about drugs, we're thinking about doses of drugs, whether it's in milligrams or milliliters or fluid ounces or whatever, going into bodies. And we can think about bodies in terms of their weight, how large they are, and often especially how much water they have within them for that dose of drug to dissolve into. So just merely based on differences in body size and body water composition, we'd expect the woman to get a stronger effect from the alcohol than the man. Now, there are actually some other reasons why women tend to have stronger drug effects from alcohol than men do typically, and I'll get to those in a later lecture. But hopefully that's just a quick illustration for you of one of the aspects of pharmacokinetics, that is dosing. So there are actually different methods that we can use to dose drugs into a body. We sometimes call these different methods of administration, and they're worth talking about in part because they involve different patterns of absorption of drug into the body. So it's not the case that once you take a drug, it's instantly available to your body in equal quantities at all parts of your body. Rather, it's the case that when you take a dose of drug into your body, it will begin to absorb into the tissues where you've applied the drug and will begin to affect the body at different rates depending on how it's being used. So how a drug gets into your body, the rate, that is to say the speed with which it gets into your body, has to do with the concentration of the drug that you're using. It has to do with interference from other substances which are present in your body. So for instance, if you're administering a drug by taking it into your mouth and swallowing it, as of course many people do with many different types of drugs, there can be interference from other substances like food that may be in your stomach during the time that you're taking the drug. And then the surface area of the membranes of your body that the drug is applied to will affect how quickly it is absorbed. So if you sniff a drug into your nasal passages, or inhale it into your lungs, or take it into your mouth and into your gastrointestinal tract, all these different ways that you can apply a drug to your body will have some influence on how quickly the drug will be present in your bloodstream, how quickly it will begin to affect the tissue of your nervous system, and how strong and how quick those effects from the drug can occur in your body. So in the next few slides, I want to talk about some of these different methods or routes of administration, and hopefully highlight for you some of the differences that occur, or that we can see in the rates of absorption and ultimately the rate of effect we get from these drugs. Okay, so the first way that I want to talk about is just the oral route of administration. This is probably the oldest way of taking drugs. This is just putting the drug, whether it's a fluid or a pill or whatever, into your mouth, chewing it up and swallowing it, and allowing that the chemicals in the drug, the different molecules that are the drug, will absorb into your body through your gastrointestinal system. It's worth noting, of course, that we have some natural defenses as animals built into this method or this route of administration. Depending on the drug we're taking, we might gag or even vomit if we take too much of the drug, or too much of the drug too quickly. That's not always the case, but it can be. Any of you who've consumed way too much alcohol too fast and have thrown up as a consequence are aware of this very obvious natural defense mechanism. It's a way that our bodies, like the bodies of other animals, have evolved to try to protect ourselves a little bit from accidental poisoning. It's also interesting to note that this method of administration involves some selective absorption, where some drugs will be absorbed relatively quickly through our gastrointestinal system and some drugs relatively slowly. So the basic idea here is that this is one way we get drugs into our body, but this way has some interesting characteristics to it that influence how quickly or how slowly a particular drug will get in. That said, generally speaking, this is a slow way to get drugs into your body. Usually it takes minutes or sometimes even hours, depending on the concentration of the drug, before effects will be observed, especially subjective psychological effects. This is in part because there's typically relatively slow absorption through the gastrointestinal system as compared to through some other routes of administration. Now, that's not always true. There's certainly some drugs that can be pretty quickly absorbed through the stomach and intestines, but relative to other methods of administration, this is often kind of a slow way to get a drug into your body. You probably know this already if you think about it. If you drink a cup of coffee, if you take an aspirin or an ibuprofen painkiller, if you take most other drugs through your mouth and swallow them, you're aware that it typically takes minutes or even longer before whatever that drug is begins to affect you. It's also worth saying that this method is generally fairly inefficient. For many drugs that you take this way, only a small fraction of the actual chemical is absorbed into your bloodstream and goes on to affect your nervous system. Some of it, depending on the drug, even a lot of it isn't absorbed and is just passed through your body. So relatively slow, relatively inefficient way of getting drugs into the body. And as I said just a few slides ago, it's a relatively selective way of getting drugs into the body. Some drugs get broken down fairly quickly by the acid environment in your stomach. So alkaline drugs, that is drugs which are chemically like bases, things like heroin and cocaine, typically don't get absorbed very well in your stomach because the stomach acid will neutralize their chemical structure. So if you had some cocaine, you could, if you wanted to, try to swallow it in your mouth and get some of it into your body that way. Likewise with heroin. It's not a very good way of getting the drug into your body because a lot of the drug will be lost, will be rendered chemically unavailable to your body that way. In contrast, drugs which are either acidic, like aspirin's an example of a drug which is chemically like an acid, or which are lipid soluble. Alcohol is a great example of this. It absorbs well into fatty tissues and also can dissolve well in acidic environments. Those type of drugs absorb very quickly through your gastrointestinal system. So if you drink alcohol on an empty stomach or take aspirin on an empty stomach, you'll get the effects of those drugs fairly fast. So again, this is just one way of getting drugs into the body, through the gastrointestinal system, or through the oral passage in the gastrointestinal system. It's relatively slow, it's relatively inefficient for most drugs, and it's also relatively selective. Now the next way we can think of, or another way we can think of getting drugs into the body, is through inhalation. And this is pretty obvious if you've yourself have ever smoked cigarettes or smoked marijuana, or know people who have. This is just vaporizing a drug, drawing it into your lungs through your nose or through your mouth, and whether you know it or not, what is happening is the drug which is vaporized is contacting the alveoli, the little air sacs in your lungs, and depending on what the drug is, it's absorbing through the membrane in the alveoli and getting into your bloodstream and circulating throughout your body. Now, just as the oral route of administration has some natural defenses, there's an obvious natural defense for smoking, and that's coughing. For those of you who've smoked cigarettes, you probably remember the first time you tried to smoke a cigarette and you coughed quite a lot. That might be true also for you who've smoked marijuana. This is your body's evolved defense mechanism to avoid being accidentally poisoned by a drug which is vaporized or aerosolized. Interestingly, and this is a little sidebar here, if you smoke marijuana, or if you know people who smoke marijuana, you may have heard of the idea of using a water pipe, or more commonly called a bong, to smoke. This is a device in which marijuana is burned, and then the smoke is drawn through a little pipe that goes and bubbles underwater, and then you inhale a mixture of cooled smoke and water vapor up through the main passage of the water pipe. Some people who favor smoking this way will argue that smoking with the water pipe somehow purifies or filters the smoke. That's not true at all. Smoking this way doesn't in any way clean the smoke. It just temperature-wise cools it and adds water vapor, which makes the smoke easier to draw into your lungs. So if you use a water pipe this way, or if you smoke this way using a water pipe, you'll probably be able to draw longer breaths or longer hits from the marijuana smoke than you would otherwise. The same goes if you're using a tobacco hookah pipe this way. So ironically, although some people talk about it being a safer way to smoke or a cleaner way to smoke, it actually can be a little bit of a messier way to smoke from the perspective of your lungs, because you decrease your coughing reflex and can actually draw a lot more smoke into your lungs. So just one kind of sidebar talking a little bit about the inhalation route of administration. Another interesting sidebar is within the last couple of years, I've seen advertised on the internet and elsewhere alcohol vaporizers. And this is a pretty simple idea, in fact something that you probably could have come up with yourself if you were sufficiently bored or hanging around your apartment, and maybe you have one of those water vaporizers that you plug in when you have a bad cold to make the air more moist to help your sore throat. You can buy devices, or again you could probably make one yourself, that will vaporize alcohol. That is to say, heating the alcohol in your liquor, like your bottle of vodka or your bottle of rum, to a temperature that is high enough that the alcohol molecules begin to go into gaseous form. They go from a liquid to a gas, creating a mist of alcohol that you can then inhale yourself. People who do this can get fairly drunk from it fairly quickly. In principle, it should be a good way of getting alcohol into your body for reasons that I'll get to in just a second, or in a slide or two more from this. I've heard from having read some reviews of these devices that in a way they're kind of wasteful because it's hard to really gulp in as much mist of alcohol as you might want to get drunk. And oftentimes if you just want to get drunk, it's easier just to drink the alcohol because, as I've already said, you can absorb alcohol pretty quickly through your stomach. But there's certainly enterprising young people out there, or maybe not so young people out there, who try to get drunk this way, vaporizing alcohol, and it's just trying to take advantage of another route of administration into the body. So, as compared to the oral route of administration, the sort of inhalation route of administration is relatively fast. For most drugs, it takes only seconds for the drug to become available to the body. That's because the alveoli in your lungs, although they are individually rather small, collectively have a very large surface area. So there's a lot of space for the drug that you're inhaling in the smoke or vapor to contact with those membranes and enter into your bloodstream. And of course, your lungs are designed by evolution to be very efficient at gas exchange. That's how you breathe and how you oxygenate your blood and how you get rid of carbon dioxide. So your lungs are designed to get most molecules in and out of your bloodstream very quickly, and we can take advantage of this by administering drugs that way. If we do, for most drugs, we get relatively fast absorption and fast availability of the drug in your body. Any of you who've smoked nicotine or smoked tobacco to get nicotine or smoked marijuana are probably aware that you get a strong effect from the drug pretty fast, within seconds, as compared to within minutes or longer for an oral administration. As I've already said, this is a relatively speaking efficient way of getting drugs into your body because as long as the drug can be vaporized, and as long as it can be absorbed across the membranes of your lungs, which many drugs, indeed most of the drugs we're interested in, can, it's almost immediately available in your arterial blood, and so it can almost immediately get to your nervous system and start to give you the effects that you want, or that you're looking for when you are smoking. So the oral route of administration, the inhalation route of administration. A third one I want to talk about, which kind of goes along with the first two, is absorption through tissues in the nose or in the oral cavity. So more simply, this is sniffing a drug or dipping, which is to say putting the drug in your mouth and allowing it to absorb through the tissue in your mouth. So sniffing a drug is familiar to us from images of people using cocaine or people using amphetamines or methamphetamine. This is where you crunch up a drug and you into a powder and inhale it through a straw or rolled up piece of currency into your nose and allow the powder to interact with the moisture in your nasal passages, become a liquid, dissolve into liquid, and then absorb through the tissues in your sinuses. Dipping, this is where someone takes something like chewing tobacco, sticks it under their tongue or in the back, sort of between their lower jaw and their cheek, and allows the nicotine or other chemicals in the tobacco to absorb into their bloodstream. This is a relatively quick way of getting drugs into the body, allowing, or at least drugs which are water soluble, because you have a lot of space in your nasal passages to absorb molecules. This is in part how you're able to smell anything. You've got a lot of space, a lot of very sensitive receptive membranes in your sinus passages that allow you to draw chemicals from the environment into your body, so you can sense you have olfactory sense of the world around you, and obviously your mouth and the upper portion of your throat have a lot of tissue there as well, so a lot of space for stuff to absorb into your bloodstream. I was tempted to skip this slide, but I just couldn't resist. There's actually another way that's related that people absorb or try to absorb drugs sometimes, and this is through enemas. This is where you take a drug, at least a drug which is water soluble, and you put it in an enema, which is a bag and a hose, which you then use to squirt water into your into your rectum, basically. This has been in the news, and it's hard to do this without laughing, but this has been in the news a few times over the last few years with the really unfortunate title of butt chugging. There have been a couple stories about fraternities where people have had contests where they've tried to put alcohol into enemas and squirt it into the rectum of new fraternity members. I was actually, believe it or not, once in a record store in Florida, and I overheard a conversation between two people talking about injecting alcohol into themselves this way. And I was really tempted to walk over to them and say, look, I'm sorry for eavesdropping, but you shouldn't do this. This is actually relatively dangerous. Drinking a lot of alcohol, just in the more conventional way, can be very dangerous too, as you hopefully already realize. But whereas if you drink a lot of alcohol, you sort of take a lot of alcohol into your mouth and swallow it into your gastrointestinal system, you can of course vomit it up, and you will if you drink enough of it. You don't have as much of a reflex at the other end of your gastrointestinal system in your rectum, and you also have a surprisingly large amount of tissue there that is very good at absorbing chemicals. So there's a lot of tissue that will absorb molecules from alcohol, in this case, into your body if you use an enema, and you can easily overdose yourself on alcohol this way. And again, just through the last couple of years, I've noticed, or students in my class have directed me to links that they've found online for people who've actually got rather sick from alcohol poisoning from attempting to use alcohol enemas, or again, more commonly and more ridiculously called butt chugging. So please don't do that. If that holds any temptation for you, please resist. Anyway, moving on, whether we're talking about nasal passages, oral passages, or nasal tissue, oral tissue, or even rectal tissue, this is a relatively fast way of absorbing drugs, at least drugs which are water soluble, and almost all the drugs we're talking about in this class are. It's usually only a matter of seconds before the drug can be absorbed into the body, especially if you're sniffing the drug. It depends, of course, as always, on the concentration of the drug that we're using. It's, for our purposes, we could say this is often almost instantaneous absorption. This is usually a fairly efficient way of getting drugs into the body, because almost all of the drug is almost immediately present in the blood supply and can be available to the nervous tissue and can start to have the effects that you want to get from that drug, whether it's cocaine or whether it's nicotine in chewing tobacco or any way like that. So the fourth way I want to talk about, the fourth method of administration, is injection, which is pretty obvious. Although I will say one thing that maybe people don't realize is what we're talking about here is injection into veins, so intravenous injection. People rarely inject blood into their arteries because there's usually a fair bit of positive pressure in your arteries. Your heart is pushing the blood around your body. So if you were to try to inject blood into your artery, it would be relatively difficult to do that. You would probably have some blood spurting out, and you might lose some of the drug that you're trying to inject. When people inject drugs into their circulatory system, they almost always inject it into the vein, which is where the blood is returning from its to the heart to be recirculated. There's less pressure there. It's easier to get the drug to go in. More rarely, people will sometimes inject drugs just subcutaneously. That's just under the skin, but not into a particular vein. I'm not talking about that so much right now. If you are injecting blood or injecting drugs into your vein, this is obviously a very direct route of administration. Obviously, or I hope obviously, your body has no natural defense mechanism. So part of the reason why it's relatively easy for people to overdose on drugs like heroin, which are sometimes injected into the veins, is that there's no way for your body to get rid of the drug once too much of it is there. If you drink too much alcohol, you can still do yourself a lot of damage, but your body can try to vomit up some of it. If you sniff too much cocaine, it can do you a lot of damage, but your body can try and sneeze some of it out. Or if you inhale too much tobacco smoke, your body can try and cough to clear its lungs. If you inject too much heroin, it's all over, or at least it's all over unless you get some good medical attention, because there's no way for your body to get rid of that drug. Relative to the other routes of administration that I've talked about, this is probably the fastest way to get a drug into your body. There's pretty much immediate availability of the drug, it is by definition in your circulatory system and will almost immediately be available to your nervous tissue to start to produce some of the effects that you're hoping for. In a sense, this is instant absorption, because all the other methods I've talked about so far, all the other routes of administration, depend upon the drug being in contact with a membrane, dissolving across that membrane and getting into your blood system. Here, the drug is instantly available in your blood system, in your circulatory system, and can, as I've already said, start to have some of the effects that you're hoping for. Again, so very fast, very efficient, immediately present in the blood. Hopefully that's pretty obvious. So the important idea here is that when we're thinking about pharmacokinetics, a big part of this is how does the drug get into your body? And by get into your body, we almost always mean get into your blood supply, your circulatory system. A lot of that has to do with the route of administration, whether you're inhaling or ingesting or sniffing or chewing or whatever else. And part of the reason that these different routes are different in terms of how quickly the drugs get into your body is that they have different defense mechanisms, and they have different membrane structures. So the membranes in your nose, the membranes in your stomach and intestine, the membranes in your lungs, differ in terms of how permeable they are to drugs, and they differ in terms of how much surface area there is, and they differ in terms of how perfused they are with blood, how much blood flows to those particular areas. So again, why does inhaling tobacco smoke lead to a very quick nicotine effect in the nervous system? Well, it's because nicotine is water soluble. Your lungs have a very large surface area. Your lungs are very good at absorbing molecules, and your lungs are very well served by your circulatory system for obvious reasons. So drugs can very quickly get into your body through that route. Hopefully that's fairly clear. So once drugs get into the body, they start to distribute. That is to say, they start to spread through the body and can have the effects that we're looking for on the nervous system. Obviously, this is a psychology class, so we're really interested principally in the effects that different drugs have on the nervous system. When we study pharmacokinetics, we sometimes talk about bioavailability of the drug. That is, how much of the drug gets to the nervous system and can have the effect that we expect it to have. That's in part because as soon as a drug gets into the system, it will start to be broken down by the body almost immediately. It depends on the drug we're talking about. Some break down faster than others, so the bioavailability of a drug of one drug can be quite different from the bioavailability of the other. Another thing that can affect the distribution of a drug is how lipid soluble the drug is. That is to say, how well it's able to dissolve through tissues in your body which have fat content in them. Now, if you've taken a good biology class or even a good psychology class, you probably are familiar with basic cell structures, or the basic structure of cells. And you should remember that the membranes of cells are made of a bilayer of lipids. That means that drugs which can dissolve well in lipids, that's to say fats, can get into cells, can get into tissues of your body more quickly than drugs which aren't particularly lipid soluble. So again, drugs that are lipid soluble, a drug like THC, which is one of the active chemicals in marijuana, is quite lipid soluble, so it can get into tissues of your body relatively quickly and can actually stay there quite a long time, as compared to drugs which are not quite as lipid soluble. Alcohol is another example of a drug that's fairly lipid soluble. It's interesting. It's both water soluble and quite lipid soluble as well. So alcohol can get into your body fairly quickly. Lipid solubility is an important issue for the blood-brain barrier. This is a system of membranes that separates the circulatory system outside of the central nervous system, especially the brain, from the circulatory system inside the central nervous system. So outside of the central nervous system, your circulatory system is, for the most part, the blood which circulates through your arteries and veins, of course pumped by your heart. inside your central nervous system, the circulatory system is a set of ducts that pass extracellular fluid in your spinal cord and throughout different parts of your brain. And molecules which are in your blood circulatory system can diffuse across the membrane into the brain or the central nervous system's circulatory system. But in order to do so, they have to pass this blood-brain barrier, and the blood-brain barrier is fairly selective about which molecules it allows to pass over into the central nervous system, especially into the brain. In this way, it's an important protective system that helps the brain to keep itself safe, at least from some types of molecules which may be damaging or disruptive to its structure or to its function. So again, the blood-brain barrier regulates what types of molecules get into the brain, and drugs that are lipid-soluble usually distribute faster into the brain than drugs which aren't. So a good example of this would be the drug heroin. I'll talk about this in a later lecture, but basically heroin is synthesized from morphine, but unlike morphine, well, relatively speaking, as compared to morphine, heroin is more lipid soluble, which means that if you were to inject morphine into your body, if you were to inject heroin into your body, the heroin would get into your brain faster than would the morphine. So I guess if you had two people, one of whom was injecting morphine, one of whom was injecting heroin, the person who was taking the heroin, that chemical would get into his brain much faster. And this is why heroin is a much stronger drug than morphine. It's because of its lipid solubility, its ability to get into the bloodstream and then into the brain relatively fast as compared to some of the other opiates that it's grouped with. So to quickly step back and review, there are different ways that drugs get into your body. There are different factors or variables that affect how drugs distribute within your body. When we think about pharmacokinetics, we think about how drugs are eliminated from your body. Your body almost immediately begins to break down the molecules that are in various drugs into other molecules to metabolize them down to other more basic molecules. Interesting to note, we'll cover this in future lectures, some of the metabolites of the drugs we look at have their own interesting drug effects. But whether we call it biotransformation or metabolism, this is just the process of chemically sort of breaking down drugs, which occurs again almost immediately once you take something into your body. People who study pharmacokinetics will attempt to model the rate of elimination of a drug, and there are different types of drugs, obviously, and different types of drugs will be broken down from the body at different rates. Some drugs are broken down based on the rate at which they're broken down depends on their concentration within the body. These are called first-order drugs, at least in terms of their pharmacokinetics. Other drugs are not broken down based on how concentrated there are in your body. These are called zero-order drugs. So alcohol is a good example of a zero-order drug. If you've drunk a very little alcohol, or if you've drunk an enormous amount of alcohol, the rate at which your body breaks that alcohol down is roughly the same. It's fairly steady. It has to do with the amount of a couple different enzymes that your body has available to do the work of breaking down the alcohol. And there's very little you can do to speed up or slow down that breakdown process. So it's a zero order, there's a zero order elimination of alcohol. For other drugs, the rate of breakdown and elimination will vary as a function of how concentrated the drug is in your body. Okay, so I've talked about a few kind of high points or important features of pharmacokinetics. I've talked a little bit about routes of administration, drug absorption and distribution, and then a little bit about elimination. This is a graphic here from a textbook that I used to use. It's a little bit more complicated than the material that I presented, but the basic idea is just to show, as I suggested earlier, there's a series of steps that take place whenever a drug is presented into the body. It has to do with how the drug gets into the body, how quickly it can distribute throughout the body, and how quickly the body can get rid of it. These are all aspects of the pharmacokinetics of that particular drug. So hopefully this will be at least a little bit of a review there, just looking at this image. Okay, moving on, I just want to finish this lecture by talking a little bit about drug testing. And drug testing has a lot to do with pharmacokinetics. It's an applied, in a sense, it's applied pharmacokinetics to solve a problem of detecting the presence of drugs in someone's body. And as you may already know, there are different types of drug tests that we can do. The most common methods are usually urinalysis, that is, taking a sample of urine and measuring the presence of different metabolites of different drugs in the urine. We can also do more sensitive drug testing if we draw a sample of blood, where again we're looking for the presence of either a drug itself or the metabolites of that drug, the kind of the broken down pieces of the drug in your bloodstream. And there are some newer methods of drug testing that involve using things like sweat or saliva or even hair samples that can provide us with clues for the presence of either a drug or metabolites of that drug. So again, we're testing for the drug or for metabolites. And what's interesting is that different drugs have different elimination times. That is, different times that it takes for the body to remove the drug to remove the drug, to make the drug no longer active and available to have its effects. There's for each drug there's an elimination time and also a detection time, a time it takes to not only remove the drug itself, but to remove all the drug metabolites from the body. So if you take a dose of cocaine, or if you take a dose of caffeine, or you take a dose of any of the chemicals that are in marijuana, there's going to be a period of time in which the drugs or those drugs themselves are detectable within your body. That's the elimination time for the drug. And then there'll be a detection time, which will typically be a lot longer, where we can't detect the drug anymore, but we can detect metabolites of the drug if we have the right test to do so. Here's a table from a textbook that is actually adapted from a pharmacology textbook, as you can see in the citation there. And you can just see that there's a range of elimination and detection times for different types of drugs. Some drugs like alcohol, we can detect alcohol in your body depending on how much you've consumed for a period of time that can be measured in hours, and we can detect some of the metabolites of alcohol for up to maybe a day afterwards. We can detect the presence of cocaine in your body for hours or days, and we can detect the period of we can detect metabolites of that drug for depending on the amount of cocaine you've used for either a fraction of a day or for up to a few days afterwards. Marijuana is kind of an interesting one because we can detect the actual substances, the active chemicals in marijuana in your body for quite a period of time, and we can also detect metabolites of them for quite a long time afterwards. And that's in part because marijuana is a fairly lipid-soluble drug. So once tetrahydrocannabinol, THC, gets into your body, it will linger in the fatty tissues of your body for a period of time, maybe even days or weeks or months, depending on how much you're using of the drug, where it can be detected, especially if we do something like a hair test where we can detect metabolites of that drug. But you get a sense here, just looking at this table, that there are different rates of elimination and detection for different drugs. And this all has to do with how quickly the drug, how quickly your body can break down the drug and then break down the actual metabolites or parts of that drug. An interesting issue that comes up with drug testing is if we detect metabolites of a drug in your body, we know, or we can at least suspect that you've used the drug, but it's not always clear when you've done it. However, if we're doing a blood test and we detect the actual drug itself, we can tell that or at least we can suspect with a fair degree of certainty that you've used the drug and it's been relatively recent. So again, this gives a sense that if we're doing drug testing and we're depending on urinalysis, where we're looking at metabolites of a drug, we have some ability to sense whether or not a drug has been used, but it may be difficult to detect exactly when that drug is available. When we do drug testing, we have to contend with two big problems that come up a lot of times when we do testing, and one is false negatives. This is where a test comes back negative when a drug was actually used. So you can imagine someone who has actually used a drug but wants to beat a drug test can undergo a couple different methods or a few different methods for trying to trick the test. So the test comes back negative even though it really should come back positive. There are different ways to try to substitute a urine sample. You have a friend who does not use marijuana and you try to get a sample of his urine or her urine that you can pass off as your own. Sometimes people who use drugs try to dilute their sample, if they're by drinking a lot of water so that their urine has a lot of water in it and relatively small amounts of the metabolites of the drug, and also by contamination. People try to take drugs which mask the presence of metabolites in their urine. If you've been following any of the news about drug testing in sports, especially in things like cycling, you know that there are often allegations of not only using performance-enhancing drugs, but using drugs which are designed to mask the presence of metabolites for performance-enhancing drugs. Those are efforts to contaminate urine sampling so that the test either comes back negative or comes back inconclusive when really it should come back positive. By the way, I'll provide some links online on Blackboard to advice about beating drug tests, not because I want to encourage any of you to try to beat drug tests, that would be really unethical, but also just because I think it's kind of interesting to see the kind of advice you can find on the internet for how to beat different drug tests. I at least find that kind of interesting or even amusing to read about, and I'll provide a couple links to interesting sources online. Again, not an advice for me to try to beat drug tests, but just maybe something that would be interesting for you to take a look at as well. So one problem when we do testing is the possibility that there'll be a false negative. Another problem is that there'll be a false positive. A test will come back positive when drugs were not actually used. So the test says you used marijuana or you used cocaine when in fact, truthfully, you did not. It's interesting to note that different drug tests have different rates of false positive, but urine tests often have a fairly high false positive rate. So upwards of maybe a quarter of urinalysis tests will come back as positive even if the person hasn't been using the drug. This means that those type of tests can be useful for screening, but they often mean that we need to do follow-up testing to be really sure whether or not a person has done a drug. Just because the test says the person has doesn't mean for certain that they really have. It gives us a sense of maybe if the person is likely to have used a drug, but it may be a false positive. And it's worth noting that this type of problem of false negatives and false positives comes up a lot in all sorts of diagnostic testing, whether we're testing for things like cancer or HIV or diabetes, or in this case the presence or absence of drugs in a person's system. If you've been following the news at all in the last few years, you've probably seen news stories about drug testing for welfare recipients. It's in my judgment at least, an unfortunate thing that every few years legislators in different states will propose laws wherein people who are receiving temporary assistance for needy families or other benefits which are given out by the state to people who are are poor or have lost their jobs or who have young children, etc. Laws will be proposed wherein those people have to take drug tests to prove that they're not using drugs in order to be eligible for their benefits. This comes up from time to time. One of the last times was back in 2011, which admittedly is quite a few years ago, but it was in Florida, which is a state I used to live in, and so in a way the news story kind of popped out at me and was kind of salient. The idea was that people who are receiving temporary assistance for needy families, or TANF, would be barred from from receiving any benefits if they had a positive drug test result for 6 months. And there are actually even stronger penalties if they had multiple positive tests. Like I said, that was way back in 2011. There have been more recent laws proposed in Wisconsin and in other states that I've noticed. I guess I understand why people propose these laws. I think as a taxpayer, the idea that maybe the money I pay in my taxes, which goes to fund some of these state programs, might also then end up paying for someone's drug use seems a little bit unpleasant. Like, I don't want my tax money going for someone else's drugs. I guess I get that. I understand why people might have those feelings. I also get the argument that drug testing like this might help to catch criminals or people who are using drugs or selling drugs, or maybe even just encourage people who are using drugs to seek treatment. Maybe folks who are using drugs will feel motivated to seek treatment if they know that they could get caught for using drugs, and when they get caught they could lose a benefit that to them and to their families is valuable. However, I think I'm also kind of sensitive to and persuaded by, to be honest, some of the arguments against these types of laws. For instance, I think that drug testing can serve to stigmatize people who receive welfare benefits. As if it's not already hard enough to receive welfare benefits, now you have to undergo drug testing, you have to face the potential stigma of people thinking you're a drug user, even if maybe you're not. Also, there's often a cost associated, or there's always a cost associated with doing massive testing, testing many thousands of people for in this case, for drug use. And I think that people who propose these types of laws are often unaware of or are underestimating the amount of cost involved in these types of programs. And there's interestingly some constitutionality issues that legal scholars have proposed about that are problematic in this type of testing. And as someone who studies as a scientist and someone who's kind of interested in diagnostic testing as a clinical psychologist, I think there's a big problem associated with false positives in test results if you're testing large numbers of people, including large numbers of people who are receiving benefits from states like TANF. It's also interesting to ask, is it the case that welfare recipients are more likely to use drugs than people who are not welfare recipients? And here, I won't present a lot of the data in this lecture, but if you go back to the National Survey of Drug Use and Health, you find that there is some evidence to suggest there are higher rates of use of some drugs among people who are welfare recipients than among people who are non-welfare recipients, at least based on some data from the early 2000s. But it's also interesting to note that we don't see very high rates of abuse or dependence among people who are on welfare. So it doesn't seem to be the case, at least in the National Survey of Drug Use and Health, that there are large numbers of people who are on welfare programs who are abusing or dependent on drugs. And indeed, other researchers who have used other data sources, not just the National Survey of Drug Use and Health, have found similar rates of drug use among people who are welfare recipients and people who are non-welfare recipients, and again have found relatively low rates of abuse or dependence of folks who are on welfare programs. So being aware of this research persuades me, at least, that I don't think we have a very large problem in our society with welfare recipients using drugs. So the whole idea behind going out and testing these folks seems a little bit misguided to me. It just doesn't seem to be the case that we have lots of folks who are receiving these type of benefits who are then turning around and using drugs, and even fewer still who are using them enough to really be diagnosable as abusing or dependent on drugs. As I mentioned before, testing can be expensive, and in the case of Florida, a state which actually managed to institute one of these laws for drug testing, during the period of years in which drug testing was running, it ended up costing the state a little over $100,000 per year to test people for drugs, which was much more than the money that was saved by kicking people off of welfare who were tested positive for drugs. So to the extent that folks who backed these programs wanted to save money, wanted to remove people from welfare who were abusing drugs and who in some way maybe didn't deserve welfare, the idea to test them ended up costing the state, at least the state of Florida, much more money than it saved in terms of decreasing the costs of welfare programs. It's interesting, like I noted before, there are some legal issues that come up around testing people for drugs. In the case of Florida, a Florida court ruled that testing was an unconstitutional violation of the Fourth Amendment, which, if you think back to your high school civics class or social studies class, you'll remember protects us against unreasonable searches and seizures. That court and other courts have ruled that It's unconstitutional to require people to undergo testing without any, in essence, probable cause, without any reason to think that they will be using drugs. It's unconstitutional to then mandate that people get tested for drugs. So there's some constitutional issues involved with massive drug testing. And like I said, lastly, or like I said before, I think there's some practical problems associated with testing large numbers of people for drugs. One of these is the false positive rate. Again, it depends on the sources you look at, but the false positive rates for drug testing can range as low as 5%, but maybe as high as 25% or more. And if you're testing thousands of people who are receiving government benefits, you can expect that literally hundreds of the positive tests that you get, hundreds of the positive results you get, will be false positives. And it may be difficult to know who is who, and you may be potentially telling hundreds of people that they are positive for using drugs, when in fact many of them, in fact most of them, probably won't be, which creates, I think, a huge practical problem associated with these types of programs. Now, when I do this lecture in class, I try not to kind of stump too much for my own political opinions, but it's clearly obvious, I think, in these last few slides that I'm against these types of programs. I think that some of the arguments for them are probably understandable, or at least I can be sympathetic to them, but there are many, I think, better arguments against these types of programs. Again, if I'm doing this in class, I often try to ask the question to my students, who else should get drug tested? If we're concerned about people using government benefits who might also be using drugs, does that mean we should test students who are at state-funded institutions like NDSU? All of you are receiving a government benefit by going to a university which is subsidized, at least in part, by the state. Should we test faculty? I get a paycheck from the state of North Dakota. Should I be drug tested just because I'm getting government money? Should people who get financial aid, or people who get government assistance on their mortgage or their rent, should they be drug tested? Again, I think the idea of drug testing people who get government money probably starts with sort of a good idea or in a good place, but goes pretty quickly to a bad place, something that I think most people, or at least I, would be uncomfortable with. And more generally, I think there's an important idea here that drug use creates complex problems for society. I wouldn't want to deny in any way that the idea of people who get government benefits but also use drugs is problematic. That's not a simple thing to think about. It's tricky to think about that. We often want to find simple solutions to drug-related problems, whether it's drug use, drug abuse, crime associated with drugs, etc. But we usually can't find simple solutions to complex problems. And again, I think this idea of drug testing welfare recipients, or drug testing people who get government benefits for drugs, is an example of that. Maybe an attempt, maybe even a well-meaning attempt to find a simple solution to a very complex problem. Unfortunately, an attempt that is doomed, in my judgment, to be a failure. Okay, so that's enough of me talking about my opinions. Let's talk a little bit about what you can expect in the next lecture. Coming up next, we'll have psychopharmacology, which is just, we're really going to get into the sort of the pharmacokinetics of drugs, especially the pharmacokinetics of course, of psychologically active drugs. We're going to look at how drugs affect the mind, and we're going to talk a little bit about tolerance, and also review a little bit, because we've talked about it before, about withdrawal. So that's all coming up. I hope you're looking forward to that lecture. I'm looking forward to it as well. However, until then, just step back from the computer, step away from your tablet or your device, and take a break. Allow some of these thoughts to sink in. And when you're ready, come back for the next lecture. And as I always say, thank you so much for your attention. Bye-bye.